So let's begin the session.
Okay, so welcome back everyone to session 2 of our series on RNA-Seq. Today's session again will be a bit of an introductory one where we will learn about the experimental workflow and the analysis workflow that we will be doing in RNA-Seq. The experimental workflow is mainly just to get you guys used to and like just to give you guys some information as to what goes on inside the labs, right? Even before the sequencing is done, how the samples are prepared and whatnot. The analysis workflow will be the main focus of our lecture today. And we will be getting an overview of how we perform the analysis after we get the data from the sequencing labs. Each of the steps that we have an overview of today, we will be doing in great detail in the upcoming sessions. So rest assured, you will be getting the entire package. Okay, so without further ado, I will now share my screen and we can begin. My screen is visible. Is it? Is my screen visible to everybody? Okay, I see a yes in the chat. Very good. Okay, 2 yeses. Very good. Let us continue. Okay, so today is session 2 on our series on the fundamentals of RNA-seq analysis. And our agenda for the session, as I mentioned, is the RNA-seq experimental workflow and the RNA-seq data analysis pipeline. Now, this will be the highlight of our internship. The experimental workflow, as I said, is simply to give you guys a basics of the know-how of what happens or what goes on in the sequencing labs. Okay, and a lot of you might already be knowing what happens, right? So for those of you who already know, please bear with me. Okay, and if you actually so I am a bioinformatician, right? I'm not too much of a wet lab biologist, but yeah, I try to gather as much information as possible on this to showcase to you guys. If you guys see something which I have actually incorrectly, I'm trying to inform you, please let me know. Feel free to unmute yourself and we can have a nice discussion on it. Okay, so moving ahead, a very quick brief overview of the experimental workflow, right? We always begin with the first point, which is the experimental design, where we basically define the objectives and design our assays accordingly, right? Anybody who has worked in a biotechnology lab or worked in wet lab in general would be knowing what an experimental design is, right? The second step is basically extraction, where we actually isolate the RNA from the sample and purify the RNA before we even send it to the companies or before we even send it to the sequencing lab for the sequencing run, right? The third step is after extraction, we have to prepare the library. So library preparation is one of the most key steps in an RNA-seq experiment, right? Our goal is always to enrich our target RNA, that is the type of RNA molecule we want in our analysis. We are trying to enrich that, right? Common steps include converting the RNA to complementary DNA and addition of sequencing adapters. Okay, then finally, the sequencing libraries actually go to the sequencing lab. They go into these big machines, the sequencing machines, be it Illumina, PacBio, Oxford Nanopore, whatever, and the sequencing actually happens. And you get out of them, you get these files known as FastQ files. And these files are what we take forward further and use a lot of bioinformatics tools and programming to actually perform the data analysis. Okay. So this is a general overview of how the entire experiment works. Let us first focus on these first 4 steps. Okay, how this exactly works out. So first and foremost is the experimental design. Here we basically firstly define the research question. Okay, this is where we set our goal. This is where we actually define what are we actually trying to figure out, right? We in very simple words, I should say that we are trying to basically figure out the control and the case conditions here. Okay, so what we are trying to do is it could be either a disease condition versus a healthy condition. Say for example, today I want to compare the transcriptome, or I want to compare the transcript signature of a diseased individual, say an individual suffering from lung cancer versus an individual who is completely healthy, who is devoid of lung cancer, right? That is one such example. Another example is I might want to study the transcriptome of a particular condition over the course of time. Say for example, I want to know the prognosis of a disease, say Alzheimer's. I want to know the prognosis of Alzheimer's, or I want to know the prognosis of a disease such as rheumatoid arthritis, right? Starting from its onset till late stage arthritis, I want to know what are the biological changes, what are the implications, the biological implications which are actually happening inside the person's body, right? Third example could be a basic treatment versus control. I want to figure out the efficacy of a drug that I have developed in my lab. What I can do is I can take two sets of tissue cultures of the same tissue sample, treat one with my treatment, so treat one with the drug that I have developed, and leave the other one alone, leave the other one as it is, as my control. And then eventually I can simply extract out the RNA and then figure out what are the transcriptome level differences between them. So these are just some examples. So you basically start out by defining this research question, that what is it that you are trying to figure out, what between what and what are you trying to differentiate. That is the first thing that you do. The second thing for you to consider in your experimental design are replicates. So namely, replicates are mainly of two types. The first is biological replicates, right? So biological replicates are basically the number of samples. That's it. So they are different samples, different individuals, etc., etc., and their sole purpose is to increase statistical power. The more the number of samples, the better your analysis, the more statistical significance, the more statistical power, the more confidence you have in your analysis and experiment, right? So this would mean basically, if I am studying, say, healthy individuals versus individuals suffering from rheumatoid arthritis, I will have 5 biological replicates of, say, healthy individuals and 5 biological replicates of people suffering from rheumatoid arthritis, right? So that would mean I would have 5 different people who are suffering from rheumatoid arthritis, and I would take samples from all of them, and I would have 5 different people, right, who are healthy people according to my inclusion criteria. So as a researcher, I can set my inclusion criteria that, okay, for a person to be healthy, these are the parameters that they have to satisfy, then only will I call them healthy, right? So based on my inclusion criteria, I will choose 5 healthy people, and based on my inclusion criteria, I will choose 5 diseased people, or in this case, for example, rheumatoid arthritis people, and they become my biological replicates. Okay. Technical replicates are basically the repeat sequencing of the same sample. So out of these 5 and 5, so 10 samples, what I do is, instead of sequencing each sample only once, I sequence each sample 3 times. Okay. Again, this is just so that we can increase our confidence. Okay. There can happen certain things which are one off, right? So maybe when I was sequencing sample number 3, something happened, and my sequencer itself broke down, right? Or there was a problem with the internal chemistry of my sequencing, and I did not yield as much data for that sample. So it is always a good idea to have technical replicates as well, where you repeatedly sequence the same sample, just to make sure that you are getting every nook and cranny of the information that you are actually paying for. Okay. And yeah, so these are the two main types of replicates that we should consider. Usually all of these things are set well before the group or the individual even starts beginning or even begins with their experiment. Okay. The next thing that you have to consider is the sample size, namely of the biological replicates. That is, how many biological replicates do you want per group, right? For example, a few minutes back, I mentioned 5 people having rheumatoid arthritis and 5 people who will be healthy people, or my control group, basically, right? So that means that I am using a sample size of 5 plus 5, which is equal to 10, right? Or a sample size of 5 per group, right? And okay, so there is no such number that is like, okay, like this is the golden, that this is the magical golden number. If you have this many number of samples, you're perfectly all right. There is no such number per se, but usually anything greater than or equal to 3 per group is considered okay. Okay. If it is less than 3 per group, it becomes a bit difficult. Think about it, all of you would have done titration, right? The experiment where that clear solution eventually turns pink, right? And every time our professors used to tell us that, okay, you have to perform the experiment 3 times and you take the concordant reading. What does that basically mean? I could also take the reading 2 times, right, and take an average of the two. That could also make sense, but no. What could happen is, if if you're taking only 2 observations per group, if both observations are very varied, if they have a high variation among them, the third observation actually helps you understand which of the first 2 observations is actually closer to the truth, right? If you have 3 observations where first observation is 3, say the number 3 for example, just imagine this: first observation is 3, the second observation gives you a value of 9, right? But the third observation gave you a value of 5, right? Now if you were to stop at the second observation, you don't know that which one is more correct, right? But since you took the third observation which gave you a result of 5, now you have 3, 5 and 9. Now it makes more sense that, okay, 3 and 5 are closer together, so maybe a value in and around that region is more closer to the truth. So 9 is probably an outlier in my experiment, right? 9 is probably something that happened due to some sequencing error, some technical error, or even my mistake while preparing the sample, etc., etc. There are n number of things that can go wrong in a biology experiment, in a chemistry experiment, right? So it is always advisable to have at least 3 samples per group. The more the better. Okay. The fourth thing that you have to take into consideration are batch effects. So these are effects that arise due to things which are not related to the biology at all. Okay. Say for example, today you extracted blood from 5 people having breast cancer, and you extracted blood from 5 people who are healthy, right? Now if you end up sequencing all those 5 samples as a single batch on the same day, right, who of the people having breast cancer, and then 2 days later you sequence the samples of the healthy people on the same batch, right, it could so happen that when you actually perform the statistical analysis, the differences that you end up catching are actually not related to the biology but more related to the fact that you sequence them in separate batches, right? So, and this is something that is very common in biology experiments, especially in sequencing experiments, right? That is why it is always advised that please randomize these batches, right? So for example, in the first batch I will randomly choose 2 of my control samples and 2 of my case samples, right, to actually or 3 of my disease samples to actually run my sequencing, right? And then on the next batch, when I have to run the next batch, I will take the remaining, again 3 of my control samples and 2 of my disease samples, right? So this randomizes the thing, right? Now my batch effects are not confounding with my condition, right? So now what is happening is I can actually use certain statistical techniques, or I can use um, error or I can use certain error reduction techniques to actually remove these batch effects and focus on only the biological difference. We will understand how all of this is done, of course, but on the wet lab end, on the part of the experimenter, on the part of the person who actually prepares the samples, it is very important for them to avoid batch effects by actually randomizing sample prep. Okay, so that your conditions are not exactly differentiated by the batches. In batch 1, randomly run 3 out of the 10 samples, that is fine, but you as a user should not know what are these samples. Okay, that is how you truly reduce batch effects from your end. Okay. uh, After this, of course, there are certain tools and statistical analysis frameworks that help you reduce these batch effects, but on the wet lab front, this is something that it is advised, it is one of the best practices, right? The fifth thing that we should take into consideration is the sequencing depth. The sequencing depth simply means how many reads you are generating per sample. Now, of course, before you have even done the sequencing, you have to actually decide on this. So this is kind of like a hypothetical number that, okay, this is the sequencing depth I want, means that per sample I want this many number of reads, say 20 million reads or 30 million reads I want per sample. You set this and you actually alter the biology, and you actually, sorry, not the biology, you actually alter the chemistry in terms of the sequencing reagents, in terms of the sequencing run on the machine, etc., etc., to achieve this, right? Finally you will maybe not end up getting the exact number of reads or the exact sequencing depth that you had asked for, but the number will be pretty close. If you have asked for a sequencing depth of 20 million, you might end up getting somewhere something within the range of say 15 to 25 million, but that is fine. That is still fine. Okay. And the sixth and final one is the metadata. That is, you can also choose to collect the clinical or phenotypic info in alongside the sequencing, right? So for example, for people having breast cancer and versus people who are healthy, you can choose to collect other information such as, okay, if the person is having breast cancer, you can choose to collect phenotypic information about the tumor, the size of the tumor, the radius of the tumor, how smooth it is, the curvature of the tumor, etc., etc., right? So again, these are all metadata, and these are all what will help you actually correlate your biological results, correlate your RNA-seq analysis results with the actual phenotype, and actually give you a greater confidence as to whatever you have analyzed on the molecular level, is it actually showing some kind of trait or phenotypic effect, right? And that is why collecting metadata is also so important. So please give me a show of hands if you have understood what experimental design is all about. Give a few thumbs ups, maybe, if you can. Yes, anyone? Okay, I see one thumbs up. Others, any questions until now? Okay, quite a few thumbs ups. Okay, then we are good to go. Okay, let's continue. The next part is sample collection and RNA extraction. So remember, we are going to be doing RNA-seq. So of course, we will be extracting RNA, right? So your sample correction and RNA, sample collection and RNA extraction is a very important step, because high integrity RNA is crucial for your experiment. Okay, if your RNA quality is bad, your sequencing is getting done on bad quality RNA, the data generated is already bad quality, you cannot trust any results you get from the downstream analysis pipeline. Okay, sample sources could be anything, starting from cell cultures, it could be tissues, it could be bodily fluids such as blood, saliva, it could be organoids, etc. Anything from which you can extract RNA, that could be your sample source. Okay. Now, RNA extraction, I'm pretty sure quite like a few of you might even know better than me how it is done. But as far as my very limited knowledge goes in this field, it is usually done using methods such as the trizol method. It can be done using column-based kits, or it can be done using magnetic beads. Okay. I'm pretty sure there are several other standardized methods as well. I'm not really too aware of all of these, but yeah. So this is just like for the sake of just saying I'm telling you this. I'm pretty sure all of you know this much better than me. Anyway, the other thing that we must make sure is we must make sure that our RNA that we have extracted is pure, and we do this by actually measuring two quantities basically. First thing that we measure is something known as the A260 to 80 ratio. Okay, so that is basically nothing but the absorbance of our sample of our RNA at it is a ratio of the absorbance of the RNA at a wavelength of 260 nanometer divided by the absorbance of the RNA at a wavelength of 280 nanometer. Okay, the most ideal value for good quality RNA is around 2, right? So anything less than 2 would actually mean that you might have some protein contamination there, and anything greater than 2 actually means that you might have some excess DNA contamination in your sample. Okay, and that actually makes sense, because if you think about it, proteins absorb absorb UV radiation the best at 280 nanometers, right? So a ratio where 280 nanometers absorbance is the denominator, you want a higher value, you don't want lower values, because that would mean that absorbance at 280 was higher, and that would automatically mean that your sample actually has more of protein than of nucleic acids, right? And similarly for 260 is actually the ideal wavelength of UV light which is absorbed by nucleic acids. So that is why you need something which is around 2. Anything more than 2 again would mean around that would mean again DNA contamination. Okay, and that mainly comes down to the fact that RNA is ribonucleic acid and DNA is deoxyribonucleic acid. So basically DNA lacks 2' hydroxyl group, okay, at the 2' carbon. So that is why the absorbance spectra kind of changes. Okay. The other thing that people usually measure in the lab is something called the RIN, or the RIN, which is the RNA integrity number. Okay, the lab that I worked in, mostly they used something called the Agilent bioanalyzer to actually measure these things, to measure this, and a RIN number of greater than 7 is generally considered to be good enough for an RNA-seq experiments. Okay, so with all of this, I have one question to you guys, that is, why do you think degraded RNA, so RNA which has been degraded, by that I mean RNA which has been degraded, so by that I basically mean that RNA which has been basically chopped off into various small pieces, right? So such types of RNA, such type of degraded low quality RNA would actually affect our sequencing results. Why do you think that might happen? Any guesses? Anybody? You can unmute yourself and take a shot. Okay. Not too many, not too chatty of an audience here. Okay, no problem. We can move ahead then, right? So the answer is technically because degraded RNA produces shorter fragments, which leads to biased representation of transcripts favoring the 3' ends. Why exactly favoring the 3' ends? You miss out on information on the 5' end. Why exactly that happens? It will become clear in the next slide. Okay. So the next slide is all about library preparation. So what we have done till now is we have decided on our experimental design. We have extracted the RNA from our sample source and we have purified it. Now it is time for us to prepare the library, right? So the first step in library prep is your mRNA enrichment or rRNA depletion, based depending on the type of experiment you are running. Okay, if you want to investigate only coding RNAs, that is RNAs which eventually end up creating proteins, right? So you would employ a technique known as poly A selection, where what you're basically going to be doing is, all of you guys know that mRNA has a poly A tail at the 3' end, right? So all you have to do is you have to use an oligo DT, basically an oligo DT stretch, or a stretch of T's or a stretch of thymines, to basically hook these mRNA molecules onto these molecules, right? So basically we end up using some magnetic beads. What these magnetic beads have are these oligo DT fragments, and the poly A tails eventually bind to these oligo DT fragments, and we can extract out this part using we can extract out the mRNA using these oligo DT beads, right? Alternatively, you can also employ something known as rRNA depletion, which is basically depletion of the ribosomal RNA. There are certain enzymes that allow you to deplete ribosomal RNA specifically while keeping the other forms of RNA intact, right? This is mainly done when you are also trying to study not only coding RNAs but also the non-coding RNAs, right? So as we all know, in today's world, we know that non-coding RNAs play a huge role in gene regulation. So it is also worth studying them and sequencing them and performing data analysis on them. So that is when you end up using ribosomal RNA depletion techniques. Okay. And this actually answers our question from the previous slide, that if my RNA is degraded, it leads to a biased representation favoring the 3' ends. Why is that? Because if you think about it, the poly A tail is at the 3' end. Now, if this RNA molecule is depleted, that means it is cut somewhere here, cut somewhere here, cut somewhere here, etc., etc. So now when I'm using these magnetic oligo DT beads, what I'm ending up doing is I'm only picking up the region which is closest to the poly A tail, or which is closest to the 3' end. So I am losing out this information near the 5' end because it does not have that poly A tailing, right? So that is why it is not a good idea to go ahead with your experiment if your RNA molecule is already fragmented. Okay. Now, after you are done selecting the poly A RNA or whatever RNA type you want to select, right? After you have done with the enrichment step, you perform fragmentation. Okay. Here you basically fragment the RNA into smaller pieces. There are several techniques to do this. One of the most common techniques is using sonication, wherein you use sound waves to actually break down these RNA fragments into small pieces. And this is actually important for the further steps wherein you perform the complementary DNA synthesis and the sequencing, right? So the third step here is the cDNA synthesis. So the machines that we use, actually, they are not really equipped to sequence RNA directly, right? They are more equipped to sequence DNA, right? So the only way we can do that is if we can extract the RNA from the sample and then convert it to cDNA, right? So that is why we end up actually reverse transcribing the RNA that we have fragmented into cDNA, right? So we first do first strands in the, and this is how actually reverse transcription works. We do the cDNA synthesis one strand at a time, right? Then we do one of the steps that we actually saw yesterday, which is the adapter ligation. Once we have the cDNA fragments, we ligate the adapter sequences to them. And as you all know, the adapter sequences are made up of three parts, right? One is the sequencing primer, the other is the index, and the other is the actually the oligo, which helps the insert or the fragment bind to the flow cell, right? And the indices can be of actually two types. So there are single index and dual index ones. It's just nothing but they just try to make unique combinations out of it. Does not really matter if you're using a single index or dual index. It only depends on how you're designing your experiment, right? And finally, after we are done ligating, after the adapter ligation is done, we do a PCR amplification, wherein we amplify the reads, we amplify the fragments and make it up to a concentration which is enough for us to submit to the sequencer, and so that the sequencer can actually detect and carry out its job. That is why it is necessary for us to perform a PCR amplification. Now, once again, a very simple question I would like to ask you guys, just to make sure that we are all on the same page. That is, why might we prefer ribosomal RNA depletion instead of poly A selection in some experiments, right? And of course, the answer is pretty simple. We have already discussed it. Would anyone like to give it a shot? Okay, somebody answer. Okay, we do not get high quality RNA all the time. Think about it. The quality of RNA was already fixed at the step before this, right? This is, we are talking about library preparation here. You already checked the quality by checking the 260 to 80 ratio. You check the RIN number, and now the quality is fixed. That you are now starting with good quality RNA. But still, you sometimes you might prefer to do ribosomal RNA depletion. Sometimes you might prefer to do poly A selection. Why would that? Why that? Study gene regulation. That is a good answer. Yes, to understand non-coding RNA. Yes, correct. You are thinking on the right direction. Absolutely. So if you are to do poly A selection, that basically means the objective of your study is to actually study or actually understand the coding RNA. If you also want to understand and study the non-coding RNA, you would end up using ribosomal RNA depletion, because then you can preserve the non-coding RNAs as well. You can capture the non-coding RNAs as well. Okay. Any further questions? Can you please give me a thumbs up if it's clear till here? Okay. Let us go ahead. So now that we are done with the library preparation, the next step is the actual sequencing, right? So this is exactly what we saw yesterday, that how exactly that sequencing happens inside those machines, right? So once the RNA-seq libraries are sequenced, right, once they are prepared, they're sequenced using these high throughput platforms, right? So you would ideally use Illumina for short read sequencing, right, and PacBio or Oxford Nanopore for long read sequencing. And as I said, the typical sequencing depth, or the total number of reads per sample that you would want from each sample, basically the total number of reads, that is about 20 to 30 million reads per sample for any standard RNA-seq experiment wherein you are supposed to do expression analysis, or you're supposed to figure out differentially expressed genes. Okay, so this is, again, these are ballpark numbers. It's not that there are no like hard and fast rules that it has to be 20-30 million reads. I have seen people perform RNA-seq and then perform differential gene expression analysis with sequencing depths of as less as 8 or 9 million reads per sample. That is also something that I have seen, and people have actually published papers using such data, and it is absolutely fine. But these are some of the best practices, right? This is what is advised by the experts out there. Okay, and if you want to actually capture transcripts which are very low abundant transcripts, like genes which in general express very less inside these samples, it is actually a good idea to increase your sequencing depth. Because if you're increasing the sequencing depth, you're basically increasing the chances of you capturing even small amounts of expression. Okay, so again, just throwing out a question to you guys. If you feel that you can answer, please go ahead. When would you want to use long reads instead of short reads for your RNA-seq experiment? A very simple question. We have already discussed this once yesterday. Yes, any ideas? Okay, so for the isoforms, yes, correct, very good. The simple answer, as Pavitra said, when you're trying to capture full length transcripts and identify splice isoforms, that is when you would actually opt for long read sequencing using PacBio or Oxford Nanopore, or and not short read Illumina. Okay. Okay, so with that said, we will move on to the data analysis pipeline, right, of RNA-seq. As I said, this is going to be the crux of whatever we do throughout the internship. Okay, if you have any questions still here, please ask. Please unmute yourself and ask away.
Hello.
Yes, please go ahead.
Can you please explain the last
Hello, you're
part
not on.
regarding the isoforms?
Yeah, so the question was that when would you want to actually perform long read sequencing and not short read sequencing for an RNA-seq experiment, right? And the idea is that if you are trying to basically figure out splice isoforms, right? So for the same gene, right, there could be different splice isoforms. You are aware of what splice isoforms are, right?
Yeah,
Wherein,
yeah,
yeah, so for the same gene, if there are different splice isoforms, you would actually want to what can I say, you would actually want to capture the entire gene body as a whole and not in very small fragments, right? To actually figure out that, okay, say for example, for gene A, if there are four types of splice isoforms available which actually trigger different biological functions, not an overall different biological function, but slightly different biological function, right? Now, if you are to quantify that, right, it would not make sense for you to actually break down the entire gene body of gene A into very small fragments and sequence them and then try to stitch them together. You wouldn't know which exon goes with which exon at what time, right? But if you sequence a longer fragment using a long read sequencing technology, you know the abundance of isoform A with respect to isoform B, with respect to isoform C, and so on and so forth. Right? That is why you would prefer a long read sequencing technology when you're trying to understand splice isoforms, when you're trying to go even deeper into the gene body. Does that make sense?
yeah. Now it's clear. Yeah, thank you.
Okay. Any more questions, guys, up till now? Okay. Then let us move ahead with the data analysis pipeline. So as I have been saying, This is the most important part, because each and every step that you see here, we will be doing hands-on. We will be going into the nitty-gritty details of all of these. Okay, we will understand how it works computationally. We will understand how the math works behind it, everything. So it is very important you guys understand this very well. Okay. So this is a typical RNA-seq data analysis pipeline, or I should say a typical differential gene expression analysis pipeline. Okay. So what we do is we start with the raw data, that is the raw FASTQ files, right? You can either download them from various databases, which I will be showing to you guys in one of the upcoming sessions, right? There are several databases for you to download the data. And as a matter of fact, as part of your mini projects, you guys will be performing data download from these databases directly. Okay. The other way that you can do is you can extract the data from your sequencing laboratories, right? So if you've given the samples to if you have outsourced the sequencing to an outside sequencing laboratory, you can actually extract the data from there. If you have an in-house sequencing facility, you can extract the data from there, and so on and so forth. Okay. So what you will end up with are basically two FASTQ files per sample. One is known as the forward FASTQ file and the other is known as the reverse FASTQ file. In some cases, you might also end up with only one FASTQ file per sample, right? In that case, it is known as single end sequencing. In single end sequencing, we only sequence every molecule. We only sequence each molecule in one direction, that is from the 5' to the 3' direction, right? In paired end sequencing, we sequence the same molecule two times, once from 5' to 3' and then again from 3' to 5', which is what you guys saw yesterday in that Illumina video, if you guys remember. If not, please go back to that video. It's a YouTube video that has been posted by Illumina themselves. Go ahead and check it out. The sequencing that you saw there is known as paired end sequencing, right? And it is always a good idea for your RNA-seq experiment to perform paired end sequencing for the purpose of having greater, higher confidence. Okay. So in most cases, you will end up with two FASTQ files. One is the forward FASTQ file, which will actually have the sequences from 5' to 3', and one is the reverse FASTQ file, which will have the same sequences but read from 3' to 5'. Okay. So it typically, when it comes out of these facilities, out of these sequencing facilities and these machines, it comes with basically, as say for example, some sample ID underscore R1 and then some sample ID underscore R2. R1 is usually the forward FASTQ and R2 is usually the reverse FASTQ. Okay, that is how you will usually get it. Now, once you have obtained these FASTQ files, two FASTQ files per sample, what you will do is you will perform a quality control check. Okay, so whatever data has been generated, right? We will again understand how and what it basically is. But basically, it is very important for you to make sure that the data that has been generated by these sequencing machines is high quality data. Because as I said, if the data quality is bad, your downstream analysis will not be good. Your downstream analysis will be performed on bad quality data, and you cannot trust any results you get from such bad quality data. Okay, so you perform a quality control or a quality check, right? The tool to perform that is actually known as FastQC, right? And based on whether it is good or bad, you have a decision to make. If the quality control tells you that the generated data is of bad quality, right? What you can do is you can use tools such as Trimomatic to actually trim out the bad quality parts. Now, if you think about it, the data that is being generated is nothing but a string of letters, right? It's basically a collection of letters. It's a collection of A, T, G's, and C's, right? So you can choose to basically selectively keep only those letters or only those nucleotides which are of high quality. The rest of them you can choose to ignore. Of course, there are certain limitations and certain considerations for that. We will go into them in the upcoming sessions. But the idea is it is possible for you to actually increase the quality of your data computationally by getting rid of the bad quality data from your files using something known as trimming. Okay, so this is again performed by tools such as Trimomatic, which is something that we will do hands-on. Okay, so now again after trimming, you again do another quality check, and if it passes your quality check thresholds, if it passes, if you see the reports and you feel that okay, this is good enough quality for me to go ahead with the analysis, we go ahead and do the next step, which is mapping or alignment. Here what we are doing is we are aligning our reads to the reference genome, right? If you are studying a human disease, your reference genome is the human reference genome, right? So basically it's nothing but think about the human reference genome as being a very long thread, right? And you have small threads, which are your reads, right? That come from these FastQ files. All you're going to be doing is playing match the puzzle. Okay, you're going to be playing match the following. You're going to basically pick each of these reads and place it at the positions where you think it matches the human reference genome. Okay, that is literally and figuratively what you will be doing in the alignment step, or sometimes people also call it as mapping. The most commonly used tools are HiSat2 and STAR, out of which STAR is considered to be the gold standard. The next is something known as abundance estimation. Okay, here is where you quantify the expression of each gene in your data. Here you simply did the alignment. You aligned your reads to the reference genome. Now, using the information you obtained here, you perform something called as abundance estimation. We will see how, we'll see a brief overview of how you can do it in a couple of minutes. But the idea here is to understand how much has each gene been expressed. This is where you actually quantify it. Okay. Now, after you have done this, you will perform something known as differential gene expression analysis, where you compare each gene across different groups, right? You compare each gene, say for example, gene A across two groups. Say group A have people having rheumatoid arthritis and group B who are healthy people. You compare the expression of gene A across these two groups, and then you conclude whether this gene is highly expressed in people having rheumatoid arthritis, or is it lowly expressed in people having rheumatoid arthritis, or does it remain unchanged in comparison to your healthy control. Okay, so these are the three steps, and finally you will end up with a list of genes which are differentially expressed, that is either differentially highly expressed, or in other words we call them as upregulated genes, or differentially lowly expressed, or in other words we call them as downregulated genes. Okay, now what we will do is we will go through a brief overview of each of these steps today to give you guys a flavor of what is going to come up in the following sessions. After which, from the next session onwards, we'll start with our hands-on works and we'll actually perform all of these one by one. Okay. So any questions up to here with respect to this flow chart at least? Okay, I don't see anybody speaking up, so I will move ahead. Okay. Okay. So now the first step, as I said, after you have obtained the FastQ files, the first step
So,
is
Mahadev, kindly
yes,
hold on. There
yes,
is
yes.
a request to, I think, repeat one concept.
Okay, can you explain mapping once again? Sure. I would request you to hold on to the thought because we will go through an overview of mapping again. If you have a doubt, even after that, we can take it up. Okay. 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 So let us continue with the first step that we discussed here, which is quality control, right? Or quality check, right? So the tools which are used mostly are known as FastQC and MultiQC. So basically, this is the main tool, FastQC, which does all the mathematics and the statistics behind it, right, in the backend, and actually gives you the report. MultiQC is actually nothing but an aggregating tool that helps you aggregate a lot of reports together. So this tool you can run once on each sample and generate a report, one report per sample, right? Now a lot of the times it becomes very important if you, especially if you have a lot of samples. Say for example, if you have 10, 15, 20 samples, right? It is not the most ideal for you to look at each report separately and then note down your observations and then manually sit and aggregate them. That becomes to a bit of a difficulty, that becomes a bit cumbersome. So this guy multi-QC actually solves that problem for you, wherein it takes all the FastQC reports and generates basically an aggregated report of all the samples together. That is why these two tools are used hand in hand very well. Okay, so here basically we try to calculate some metrics such as read quality. Again, what read quality is, we will understand in one of the upcoming sessions where we actually perform FastQC and multi-QC. I will tell you how exactly read quality is assigned, what is the math behind it, right? Everything. Just a trailer of it for you guys. We use something known as Fred encoding, okay, or Fred scoring. Again, a lot of you might have heard of it, a lot of you might know what it is. So please just bear with me. We will go through all of them in great detail. Okay. So we basically try to figure out and try to quantify or calculate metrics such as read quality, the GC content of the sequence, the duplication levels in the sequence, the adapter contamination in the sequence, etc., etc. Right? And eventually, again, as I said, after quality control, if I have poor quality reads and if I have adapter sequences, I can remove them using tools such as Trimomatic or Cutadapt. Trimomatic is what we will be using during our internship. Okay, so just to give you guys an example, this is how a typical read fragment looks like, right? These are your adapter sequences, right? The red and the blue region. This is actually your sequencing read, this black region, right? So now sometimes when you're actually doing the fragmentation, right, it is not in your hands to fragment it exactly like, okay, that okay, when we are actually doing the fragmentation, we will keep only the black region and not the red and blue region. Sometimes the red and blue regions remain, sometimes adapter sequences get into your sequencing reads, right? And that is really common, and that is a major issue in these experiments. But there is no way of getting rid of them biologically or chemically, so we have to do it computationally, right? So these tools, as I said, Trimomatic or Cutadapt, actually help us identify these adapter sequences because these are universally agreed upon. Everybody knows the sequence of these adapters. We can actually look for those sequences in our data and choose to remove them. And that is how we actually filter out garbage reads such as this, wherein you can see this is a read that is made purely of adapters. There is no sequencing read fragment here, right? So this is something that is contaminating our data, so we can remove this computationally. Okay, that is where these tools like FastQC and Trimomatic help us out. Okay, and this is something that we will actually do with our own hands when we perform the analysis. Okay, everybody understands what quality control is, or at least whatever I have shown you here? Yes. Is there any threshold level or how much we trim? Yes, there are threshold levels on several of these quantities, right? There's a threshold level for quality score, there's a threshold level for adapter content, there's a threshold level for GC content, etc., etc. All of these will cover when we actually cover FastQC and multi-QC as tools in one of the upcoming sessions. I will give you the exact numbers and the industry related gold standards as to what are these exact thresholds. But yes, there are thresholds. Any more questions on quality control right now? Okay. So the next step is read alignment or mapping. So whoever asked the question about alignment, again, pay attention here. If you have a doubt even after this, we can discuss. Okay. So read alignment is nothing but you mapping your reads to the reference genome. So as you all know, every organism has a genome, right? So if you are studying a human disease, your reference genome would be the human reference genome, right? We all know we have 3 billion bases, right? So our human reference genome, if you can, just try to imagine a very large string or a very large thread, right? A very long thread. Okay, that is basically your human reference genome. Now, these read fragments are very small in comparison to it, right? The human reference genome is what? 3 billion bases. And each of your read fragments in a short read sequencing is what? Something around 100 to 200 or 300 bases, right? So it is very small in comparison to that, right? Now, it falls on you as the bioinformatician, or it falls on the analyst to actually figure out from where exactly in that long thread does that small piece of 300 base originate from, right? So what you do is you do something known as string matching. You try to match the letters, right? So if you have this huge thread here, this long thread here, and if you have this read, what you will do is you will try to match that. Okay, maybe in this region I see this sequence ACACGA, so maybe this read came from this region in my thread. Or more technically, I should say, this read originates from this region within my human reference genome. Does that make sense to you? Divina, do you understand? That is the basic idea. Okay. Now let us get into a little bit more of a technicality. Okay. So here is our genome, right? Of course, we all know the genome is not present as a thread inside the nuclei; it is present as chromosomes, right? But then again, let us, for the sake of simplicity and for the sake of understanding, let us assume it is present as a very long thread. Okay. So the first and foremost thing that we do is we break down this long thread and actually index these fragments. So consider this: if this is your genome, if this is your reference genome, you break it down into these small fragments. Okay, these are known as 𝐏𝐥𝐞𝐚𝐬𝐞. Okay, we will get to what 𝐏𝐏𝐸MERs are, don't worry. Just for now, you just understand it like 𝐏𝐏𝐸MERs are basically fragments, small fragments of a large string, right? Where K stands for the length of each fragment. Here you can see that the length of this fragment is 6, this fragment is 6. So each fragment here is basically 6 letters long or 6 nucleotides long. So these are actually 6 mers of this string, basically, right? So if you have a very long human reference genome, you break it down into small fragments and you do something known as indexing of all the fragments. Okay, this indexing is the same as the index you see at the end of your textbooks. Okay, which basically tells you which word is present on what page. You have an index right at the end of the textbook. If you're looking for a word, right, if you're looking for a very niche word, a very particular word, instead of looking for that throughout the textbook, instead of starting from the first page of the textbook and iterating over every page, you can simply go back to the index, right? You can go to that index page, look for the word there, and there the coordinates are already mentioned, right? That okay, this word has been mentioned on this page, maybe on page number 213. So you can directly open page 213 and actually see the word there. That is how indices work, right? This is exactly how indices work in bioinformatics as well. So what you do is you break down this long thread into these short threads, and each thread is assigned a unique identifier. It's kind of assigning each thread a unique address, and you make one big file that contains each thread and its unique address. Now, when you have a new read, it is not necessary for you to look through the entire genome, right? If you have a read of 100 base pairs, I mean, if you want to use brute force, what you can do, you can start from base pair number 1 and look through till base pair number 3 billion and find out where exactly you have the match. That is plausible, yes, but that is extremely computationally intensive and time taking. So the bioinformaticians and the engineers and mathematicians came up with a way where you can actually fragment the genome into small parts. Each fragment is assigned a unique identifier value, and whenever you see a good match of a read with any of these, right, you can simply look up the unique identifier value. You can look up the unique address which tells you where exactly in the genome that particular fragment comes from, And this guy, this read, will eventually match up to that region in that genome. Right? Make sense to people? Yes? Okay, I see one yes. There's another thumbs up, another yes. Very good. So this is the general idea, the basic idea. But it's not as simple, right? Just like we break up the genome, we also break up a single read, right? And there is a reason behind that. Okay. So just for now, assume this is the read that you have generated, and you break this up again into 6 mers. Okay. Now, what will happen if we don't break up the reads? Let us see this example. This is my sequenced read. Here I have broken it up. Okay. Now imagine this base, this A, was actually not in the reference genome. Okay. And that can happen due to n number of there are several versions of the reference genome that you might be using, right? So there is something called the ℏ19, there is something called the GRCH37, the GRCH38, etc., etc. Or you as a researcher, you having an independent lab might also have your own version of the reference genome. Say if, for example, if you're doing an experiment using yeast or E. coli, right? Your lab might have its own strain of E. coli which has its own reference genome, right? That is absolutely plausible. So mismatches like these are very common. Now think about it. If you had taken this whole read at its face value and you see that there is a mismatch, this A is not there in the reference genome, you would discard this read as something which is an artifact or something which is not actually part of your organism's genome, right? You would end up doing that. But since you have broken it down into these fragments, now what you can do is you can try to match these small fragments one by one to the reference genome. Now what will happen is out of this read, you made these 6 fragments, right? Out of the 6 fragments, only one did not match a region in the reference genome, but the rest 5 of them, they matched a region in the reference genome, and a contiguous region, mind you. Okay, of course, it has to be a contiguous region because this is a contiguous read, a continuous read, right? So if these 5 matched a contiguous region in the reference genome, but this guy did not, then you can actually attribute this mismatch to something such as a bad quality base call, or something such as, okay, it was just not there in my reference genome. But you are not discarding this read, you are not actually missing out on information, right? If you had not fragmented it, if you had treated this entire thing at face value, and this A would not have been part of the reference genome, you would have discarded the entire read and missing out on information, when in reality this A might just be an error of your sequencing machine. So make sense? Does this make sense that why we fragment the reads as well, right? Make sense to everybody? Yes, give me a thumbs up or a yes. Okay. Okay. So two things we learned here today, right? Yes, you have a
Yeah,
question.
I have a
Yes,
question.
yes, yes. Please go ahead.
So, if this error is due to the like sequencing machine error, if you're saying, then we are going to proceed with this sequence further for analysis or not?
No, you don't need to resequence again, right?
No,
You
no,
it
I
is.
will say,
Mhm.
will this be included in the like further downstream or not? I'm asking that part.
Huh. So your question is whether this will actually affect our downstream results, right?
I will going to take this further down to the for further analysis or not. I will going to just discard this sequence or what.
So the read, if you, as I said, if you had taken the entire read, you would probably end up discarding it. But since you broke it down into fragments, right? Now you know that it might have been actually a sequencing error. So you still take that read further for the downstream analysis, because it is just a one base error, right? And these tools and these frameworks which are created, they are made with the assumption that your sequencing machine will invariably make some mistakes.
Okay,
Okay, so
so
yes,
I will,
you
so
continue, you take
my,
take it through.
so
Yes.
I going to discard that only a nucleotide in this particular sequence, right?
No, you won't discard anything. You will take everything from here, because eventually you will not deal with the raw sequences, you will deal with the gene names. Okay, that will become clear in the next slide. Okay, when we actually assign the gene names to each of these reads. Okay, so
Okay, sir.
just bear with me for 2 minutes, you will understand what I'm trying to say. Okay.
Hi.
Yes.
I just want to know what what are the you're talking about the 𝐏𝐸𝐵, right? So I'm just thinking, like, how do you decide on the 𝐏𝐸𝐵, like which 𝐏𝐸𝐵 to choose? Like, is there an option to choose for the 𝐏𝐸𝐵, or is it?
Mostly these tools, they have default mathematical models to actually choose the most optimum 𝐏𝐸𝐵 level. Okay, a lot of high level statistics goes into it. This is only for an example purpose. Okay, people 6 MERS, 7 MERS, these are actually very small when you think about the scale.
Yeah.
Six is very small as compared to 3 billion, right? So these are only for like educational purposes, only for representation purposes. In reality, you might end up with 30 or 40 MERS. You these are parameters you can choose, you can try to modify if you want, but it is best to not modify these parameters, as these tools. So for example, for alignment, we'll be using something called star. Star has its own algorithm to choose the best k, the best value for k, in terms of what 𝐏𝐸𝐵 you should use. Okay, so it is a good idea to not mess with that.
Okay, my other question would be like,
Mhm.
I mean, for me, I think the doubt came in because it was a very small number. As you said, it's like
Correct.
6 or 7 and against a very big data.
Correct.
So does it actually like also change between the type of, like, say, for the RNA versus the DNA alignment? Like, does it change according to it, or?
Uh, all of these things are taken into consideration when you actually think about the experiment. So if you're sequencing DNA with the purpose that, okay, I will perform genome assembly, for example, right? Genome assembly is the technique wherein you actually sequence and you actually figure out the genome. You don't have a genome per se, and you actually figure out the sequence of the DNA, right? In that case, you would require a much higher 𝐏𝐸𝐵, because you would require much larger overlapping sequences. Makes sense, right?
Yeah.
So in that scenario, yes, in that case, yes, it changes with respect to the starting material, but rather I would like to reword it and say it changes with respect to your experimental design. Whether you are trying to perform differential gene expression, whether you're trying to perform genome assembly, whether you're trying to figure out variants or something known as variant calling, it will change with respect to that.
Okay.
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 Yeah.
Thank you.
No problem. Okay, so Shubhajit, hold on to your thought about whether we should discard that singular A or not. We will get back to it. Okay.
Sure.
Okay. Any more questions here, people? Okay. Let's go ahead and understand what happens after this. So now we understand that there are two levels of fragmentation happening. One is when we are fragmenting the reference genome and creating the index, right? Or kind of like creating a contents page of your book, right? And the other is fragmentation of your sequenced read, so that you don't end up discarding a read just because there are a few mismatches. Okay. Now something very beautiful happens, which is read quantification. So once you are done with this alignment step, you actually end up with a certain file known as the SAM file, or sequence alignment information file. Okay. So again, we will see what these SAM files look like, what is the structure, everything in the upcoming lectures. But just take my word for it now. You end up with something called as a SAM file, which has the information as to where exactly did your read map, right? So if you guys remember, each read, by virtue of its adapter sequence, has a unique identification number. It has a unique identifier value, right? Now what happens is each read is now assigned positions, that okay, this sequencing read has mapped to position, say for example position number 101 to position number 202. Okay, So a region of around 101 bases, this read has mapped to a region of around 101 bases, right? So we have positions from starting from position 1 till position 3 billion. Somewhere in the middle, this read would have mapped, and this tool, which is star or HiSat, etc., which performs the alignment, gives us the information with respect to the numbers that, okay, from this position to this position, this read has mapped. Okay. Now imagine you have this for all your 20 million reads in your experiment per sample. Okay. Each read, whatever position they have mapped to, you have that information in a single file that is known as the SAM file. Okay. There are a lot more other informations as well in the SAM file, and we will come to them when we actually study and understand the SAM file. But this is the key information that is there in SAM file, that is from what position till what position did this sequencing read match. Okay. Now that you have that information, you have another type of information in another type of file known as the GFF file, or to give it the full form, it is known as the gene feature format file. So here you can see this is a gene name XKR4, and for that gene name, this GFF file will have the information as to what chromosome the gene is present on, and from what position to what position this gene body runs. Okay. Now, if for each read, if you know the start and end position of the read in your reference genome, and you know the start and end position of a gene, what you can do is you can compare these numbers and actually figure out whether your read falls between these two positions, right? And therefore, if a read falls between the positions 3204563 and 3661579, that means that read can be attributed to this gene XKR4, right? Make sense, people? Does it make sense? Yes. So Shubhoji, to answer your question, we ended up not discarding the A, right? But instead we are dealing with the gene name here, because we know that this A might have been a mistake. Yes, but we did not discard the read, we kept the read. And eventually we see that this read falls under this region of XKR4. So there is no need for us to cherry pick and remove the single nucleotides at this step, because eventually we end up assigning gene names to each of them, right? Does that answer your question now?
Yes. Okay. Understand.
Okay. So now imagine doing this for all 20 million or 30 million of your reads. So here you can see the example for only two genes, XKR4 and RP1. This will be there in your GFF file for all the 20,000 genes in your genome, or mostly 20,000 genes, right? That is the number of genes we have mostly. So this information will be there for all of those genes in your genome, right? And now all you have to do is you have to check the coordinates in your SAM file for a read and check the coordinates for a gene in your GFF file, compare and figure out how many of your reads belong to this gene, how many of your reads belong to this gene, how many of your reads belong to some other gene, so on and so forth. And eventually, once you have done this using tools such as feature counts or HT-seq, you end up with something like this. You end up with this. This is known as a counts matrix. Okay. It's just a very fancy way of saying that this is a table. You basically end up with a table where the first column is the name of the gene, and every other column after that is basically the name of each sample. Okay, or your sample identifier. Okay, and each row is a gene name, and under each sample column you get how many reads mapped to that gene. So for example, for sample 1, for gene A1BG, there were 30 reads that mapped to it. For sample 2, there were only 5 reads that mapped to it. And for sample 3, there were only 13 reads that mapped to it. So on and so forth for all your samples, and so on and so forth for all your genes. So this is a table or a matrix that will have 20,000 rows for all the 20,000 genes that you have, and as many number of columns as the number of samples in your experiment. This is known as a counts matrix. Now, all of the steps we did to reach here are known as pre-processing steps. You start with a FastQ file, which basically has read information, and eventually you end up with a very structured data, or a data in the form of a structured matrix that tells you how much each gene is present in each sample. Is it clear till now for everybody? Okay, two.
Hello.
Okay, yes, you have a question. Please go ahead.
Can you explain the GFF file again? I just
Okay.
get.
So your GFF file typically has this information that you see here. For each known gene, it has the information as to what chromosome the gene belongs to and what are the positions of the gene body, right? So your gene body will consist of everything, right? It will consist of the promoter region, the exons, the introns, everything. So it will have that information that, okay, for this gene XKR4, it belongs to chromosome 1 and it starts from position number 3204563 and goes on till position number 3661579. Okay, and this information will be present for all the 20,000 genes in your GFF file. Does that make sense, Shubhajit?
Yes. Yeah. Thank you.
Okay. We will see how a typical GFF file looks. I will show it to you guys. So even if you don't understand right now, don't sweat it. We will be revisiting each and every one of these concepts again and again. Okay. Okay. So now that we have figured out how we can reach till the counts matrix, now we are going into a little bit of the statistics. We are going into a little bit of the differential gene expression analysis. Okay. So the first thing that is really important for us to understand is a concept known as normalization. Okay, very simply put, normalization is the method by which you are leveling the playing field for each and every gene and each and every sample, so that all of them have the same, so that all of them are same to your statistical framework. Okay, you don't want to basically bias your statistical analysis. That is why you do something known as normalization. Okay, so basically it's a correction method. Okay, it corrects for stuff such as library size, sequencing depth. So as I said, library size and sequencing depth are pretty loosely interchangeable terms. We know what sequencing depth is, right? The total number of reads per sample. But this is a hypothetical term which you set before the experiment even began. That is your target, that okay, I want a 20 million sequencing depth, right? But eventually, after the sequencing run, the number of reads you get per sample, that becomes your library size. So it's kind of like this is the hypothetical number that you set and this is the number you eventually ended up with. They can both be used very interchangeably. It is absolutely fine. But normalization's idea is to correct for differences in library size, sequencing depth, and something known as RNA composition. We will see with an example in a few seconds. Okay. In simpler words, each sample might end up having a different number of total reads assigned to it. That is absolutely fine, right? That could happen, right? For example, right, what could happen is a sample may have a very high number of low quality reads, right? Why could that happen? Say for example, during our sequencing run, something went wrong. We got a very high number, we got a very low number of high quality reads, right? So obviously if we have low number of high quality reads, we will have high number of low quality reads, right? If we have a high number of low quality reads, we will end up removing them during our quality control step, right? This is one such scenario. Another scenario is the sample might just have a high concentration on the flow cell, right? When we actually add the sample to the flow cell, it might just happen that we added more concentration of sample 5 than sample 2. That is also something that might happen, and these are things which are out of our control. It is not something we can control very well. So these are common things that can happen. Let us consider an example to understand this even better. Okay, consider this. Sample 1 has a total number of reads which is 635. Sample 2 on the other hand has 1270 reads, total reads, right? So clearly we can see that the total reads for sample 2 is actually double that of sample 1, right? This does not necessarily mean that the genes in sample 2 were transcribed twice as much in sample 1. As I said, any of these two things could have happened, right? For example, it could so mean that sample 2 had very few low quality reads, right? When we sequenced, maybe both of them had 1270 reads, right? That is possible. Both of them had 1270 reads. But when we used the tool FastQC to perform the quality control, the quality check, a lot of the reads in sample 1 turned out to be of very poor quality. So we ended up removing them using the tool Trimomatic, or we ended up trimming them, right? Now what happens is, downstream of our analysis, we are met with a situation where sample 1 has an overall read number of 635, while sample 2 has an overall read number of 1270. This is a problem, right? But this is a problem that is out of our control. This is a problem that we did not cause, right? And this is very common in sequencing experiments, and it will happen no matter how good of an experimenter you are. Okay, now what we can do is, we cannot take these values at face value. We cannot take these values and just perform an analysis, right? Because according to these values, this gene A1BG is twice as expressive in sample 2 as compared to sample 1, right? But for example, both these samples are coming from a healthy control subject. If that is the case, then that means there is something horribly wrong here, right? So we cannot take these at face value. Instead, what we will do is, we have to adjust these numbers. We have to adjust these counts. Okay, if you think about it, each of these numbers, if you see, is actually the exact double of its counterpart, right? So here we have 30 for A1BG and sample 1, we have 60 for A1BG and sample 2, here we have 24, here we have 48, and so on and so forth, right? So we need to adjust the read counts per gene to make up for the difference in how many reads were actually assigned to each sample. This sample had more reads assigned because there were high quality reads. This sample did not get as many reads because it had a lot of low quality reads, right? So one of the most common and simplest and easiest way to do is kind of calculate somewhat of a contribution value. So what we end up doing is, instead of taking the raw value, we take a contribution value. How can we take a contribution value? The first thing that should pop into your mind is, take a fraction, as in how much of these 635 reads is actually constituted by A1BG, right? So what we can do is, we can divide the read counts per gene by the total number of reads mapped for that sample. So if you divide 30 by 635, you will end up with something around, I think, 0.047. Okay. Now, for the same sample 2, you can now divide 60 by 1270, and you will end up with the same 0.047. You can do it right now if you want, on your calculators, on your phone, whatever you want. You can do it and check. So what does this tell you? This tells you that out of the 635 total reads associated with sample 1, the contribution value of A1BG is 0.047, right? Similarly, out of the total reads of 1270, the contribution of A1BG is again 0.047. That means in reality, the gene A1BG is actually not differentially expressed between these two samples, right? If we had taken the raw values, we would definitely conclude that in sample 2, A1BG is highly expressed as compared to sample 1, right? But as soon as we take the contribution value and not the raw value, our conclusion changes. And the contribution value is actually something that we end up using for our downstream analysis, because that removes any and all kind of bias that can come from read quality, concentration differences between samples, etc., etc., things which are out of our control basically, right? And this calculation of these contribution values is known as normalization. And the contribution value, the technical term for this contribution value in this scenario, are known as normalized counts. So these are your raw counts, 30 and 60. When you actually divide 30 by 635 and 60 by 1270, you get the contribution value or the normalized counts. Does everybody understand the concept of normalization? The importance of normalization?
Hello, I have a question.
Yes.
So, as you mentioned, if the number of reads can go back to the previous slide. Yes.
Yes. Tell me.
Yeah. So, as you can see over here, there are two samples, sample 1 and sample 2, having two different reads. So, after that trimming, if the number of reads are equal or nearly equal, does we need to have that normalization?
Okay, so your question is if the total number of reads are equal, does there need to be normalization? So first let me tell you this: the total number of reads across any two samples will never be equal. Okay, that is absolutely impossible. It is like a 1 in a billion chance that it will be absolutely equal. Even if that is the case, right, between any two samples, you don't work with two samples, right, in an RNA-seq data analysis, right?
Yeah.
You need to have, as I said, a minimum of 3 samples per group. And if you have 2 groups, that would make a minimum of 6 samples, right? So across the 6 samples, there will always be differences. Therefore, there is always a need for normalization. But in a hypothetical scenario where across all 6 samples, the total number of reads is the same across all 6, which will never happen, by the way. But hypothetically speaking, if that does happen, even then you need normalization. And
So
why?
normalization actually, like you saying, will cut down the value to a decimal range, yeah, 0.1 range, right? It's easy
You are thinking
to
on the right direction. Yes, it cuts down the value to a certain range. Yes, but the idea of normalization is to make the values comparable against each other. Right now, I cannot compare because the total number of reads are different. But if I normalize, I can now compare the expression of A1BG across sample between sample 1 and sample 2. So the idea is to make it comparable. Okay?
Okay.
Does that make sense? Okay.
Yes.
Okay. Any more questions on normalization? The basic idea of normalization, that is. We will look at the various techniques we use to normalize. This is the technique that I showed you. It is the most crude technique, the simplest of all of them. There are much more uh, complicated techniques to actually perform normalization. We will take a look at them. Don't worry.
Hello. So,
Yes.
are we going to have a discussion on how many types of normalizations are there?
Yes, we will study the different types of normalization techniques which are available. Yes, we will study RPKM, FPKM, TMM, TPM, all of these. Don't worry about it. We'll cover all of them. Okay. Okay. Any more questions? Okay. Then let's move ahead. We don't have a lot of time. I'll quickly start wrapping up. So after we are done with our normalization, now these guys are comparable against each other. Now it comes to the analysis part. Till now, all of the stuff that we have been doing is trying to prepare the data so that we can actually perform the statistical analysis. Now we try actually performing some analysis. Okay. So step 1 in almost any analysis is to basically plot your data and visualize your data somehow in terms of plotting or in terms of plots. Okay, these are done, especially in RNA-seq, it is done primarily for 2 reasons. The first reason is plotting tells us if we can actually expect to find any interesting differences, right? Number one. And number two, plotting also tells us if we should exclude any samples from our downstream analysis, right? There could be certain samples which might be outliers due to several reasons, due to batch effects, due to there being some mistakes in the sequencing run, due to there being some mistakes during my pre-processing steps, etc., etc. A lot of things can happen which might lead to some samples becoming outliers. So it is very important we identify them and remove them from downstream analysis so that they don't skew or bias our results. Okay. And the other thing that plotting tells us is if we can actually expect to find any differences in our experiment, okay, or in between the two groups. Okay. Now the human genome consists of about 20,000 genes. But for the sake of an example right now, let us consume for a moment that there are only 2 genes, right, instead of 20,000, right? If that is the case, it becomes very easy. All we have to do is assign one gene the x coordinate, the other gene the y coordinate, and we can simply plot a 2D scatter plot, right, for each sample. This is pretty simple. This is pretty simple to understand. Okay, makes sense. But consider this, we have 20,000 genes. If we are, is there any possible way for, okay, even if there were 3 genes, I could have done a 3D plot. Okay. But is there any way for me to visualize anything more than 3D? A 4D plot, a 5D plot, a 6D plot? Is there any way for me to visualize that properly? No, right? I cannot draw on a pen and paper with using a pen and paper. I cannot draw any more than 3 dimensions. It is physically not possible for us. So now we have 20,000 dimensions, right? We have 20,000 rows. Now it is not possible for us to have 20,000 axes. We can have one axis, two axes, and max to max another third axis. It is not possible for us to have 20,000 such axes, right? So since that is not physically possible, we employ a technique known as PCA, or principal components analysis. Okay. So what principal components analysis does is it reduces this high dimensional data. So it reduces the 20,000 dimensions into a very small number of dimensions while retaining the information that is present in all 20,000 of these dimensions. Okay. So a lot of complicated math goes behind this, a lot of linear algebra, right? What I want you guys to remember is, and what I want you guys to understand is, in PCA, each axis or each principal component, each axis is known as a principal component. Okay, so each principal component or each axis is a linear combination of all 20,000 axes, such that it explains the maximum possible variance in the data. Or in other words, you can imagine that each axis contains information from all of the 20,000 dimensions, such that it tries to maximize the amount of information it can gather. In very, very simple words, I don't expect you guys to understand the math behind PCA. It is pretty complicated, right? It falls under something called unsupervised machine learning. That is not something that we are doing right now, but we do use this regularly as an exploratory plot. And what I want you guys to understand is how you can actually interpret a PCA plot. You don't need to understand how actually the math works to plot it. I will teach you guys how to plot it in R, and I will teach you guys how you can actually interpret it. Okay. But just remember, PCA helps you to reduce high dimensional data. It helps us reduce these 20,000 dimensions into only 2 or 3 dimensions while retaining the information from most of these 20,000 dimensions. Okay. If that is clear, then I will quickly explain this plot that you see in front of you. Okay. Just give me a thumbs up if that is clear. What a linear combination is. A linear combination is nothing but a linear equation of n variables, right? So you know what a linear equation is, right? A linear equation is an equation where the power of your unknown variable, or x, is 1. right? Y equals to mx plus c is a linear equation. It is the equation of a line. Here the power of x is only 1. Yeah, so that is a linear equation. Yes, Shubhajit, you have a question. Go ahead.
So you said like each axis of the PCA is a combination of 20,000
Correct.
20,000
Correct.
genes.
Correct.
So there are, so when we plot this graph, there are two axes like PCA1 and PCA2. So what about the other one?
What about the other one in the sense?
Each axis, if you say one axis is a linear combination of 20,000 genes,
Correct.
then
Correct.
what about the other one, like which is on the so y-axis? If I say
so PC1, so each axis is, so basically what happens, PC1 is the one that explains the maximum possible variance. PC2 explains the second most maximum possible variance. PC3 explains the third most maximum possible variance, and so on and so forth. When I say each axis is a culmination of all 20,000 axes, it means that each axis tries to withhold as much information as is mathematically possible. And the amount of information each axis can withhold reduces as you go from PC1 to PC2 to PC3 and so on and so forth. Does that make sense?
Practically, how many plots we can do like for this PC?
So what you can do is, ideally, what we end up doing is we do a plot between PC1 versus PC2. We can also do between PC1 versus PC3, PC2 versus PC3, or we can also simply do a 3-dimensional plot, PC1 versus PC2 versus a third axis will be here somewhere, PC3. And we basically plot each of the samples here on the plot. Okay, that is something we...
Three PCA plots, like
Sorry?
three PCA are maximum possible, like PCA A1, 2 and 3.
So again, if I talk to you about the math, the number of principal components possible is equal to the number of axes. So you will have till PC 2000, till PC 20,000. But most of the information will be gathered within the first few PCs. Okay, so PC1 will say, for example, have 39% of the information. PC2 will have 37% of the information. So if you plot both of them together in this 2-dimensional plot, you're effectively explaining 37 plus 39, that would be 76% of your data. Right now, if you add PC3, which might have say 18% information, that is now 76 plus 18, that would be 94%. So using only 3 axes, you're able to explain the variance, or you're able to explain the information of 94% of the information in your data. Right? So you don't have to go till all the way to PC 20,000. That is the idea behind principal components, that a lower number of axes can actually gather a higher number of information, a higher amount of information. Does that make sense?
Yes, thank you.
Yeah. What I will do, Shubhajit, is you ping me once on Telegram. Okay, I will share a video with you that explains how principal components analysis works. Okay. Okay, so for anybody who is still confused with principal components analysis, I would suggest you guys watch this video. If you guys are okay with linear algebra, in the sense that if you guys are okay with basic matrix operations, you should be able to understand the video. Okay, we can have a discussion on how about the video on Telegram itself, so as to not waste too much time on the class. But okay, but Shubhajit, please ping me on Telegram. I will share that video. Okay. Yeah. Okay. Okay, so I will quickly explain, guys. After this, we have a couple more slides. I hope you can bear with me, or else we can also take it up in the next lecture. So that is fine. So I will quickly explain how you can actually interpret this. Okay, and that is going to be the most important part for you guys. So once you calculate the principal components, you end up plotting the samples, right? Here WT stands for wild type and KO stands for some knockout. Okay. So as you can see, the wild type would ideally form your control group, and the knockout would ideally form your case group or condition group. Okay. So as you can see, one very clear observation that is here is that these knockout samples are very close to each other. Or in other words, I can say they form a pretty tight cluster, all these knockout samples. Maybe KO4 is a bit farther away from the rest, but still, in this general area, they all lie together. So they form a pretty tight cluster. For the wild types as well, starting from WT1, 3, 4, 5, and 6, all of them form a pretty tight cluster, but WT2 is a bit farther away along the second principal component, right? So this guy is probably an outlier in your data because this is not clustering well with respect to the other wild types. This is where most of your wild types are. So you can be fairly sure that this is a correct region for the wild types in your plot. If one of your samples is so farther away from the rest of them, then that likely means it is an outlier. So this serves the second purpose that we talked about, right? That it tells us if we should exclude any samples from our downstream analysis. So when I actually perform any downstream analysis, I would actually consider excluding WT2 from the analysis. I would actually consider not using this anymore. Okay. The other thing that I told you PCA will tell us is that if we can expect to find any interesting differences, here we can clearly see the wild types cluster somewhere around minus 0.75 on the x-axis, while the knockout types cluster somewhere around 0.75 on the x-axis, right? Or on the PC1 axis, right? With respect to the PC2 axis, they seem to be near about the same region, not too much of a difference. But with respect to PC1 axis, there is a very clear difference between them. So this tells me that, okay, these are two groups which are biologically different. Because remember, this plot is built on this data, the normalized data, of course, not the raw data, but the normalized data. This plot is basically built on this table, right? And this table had all the information with respect to the gene expression. So here, what I'm basically trying to say is all the knockouts have a similar expression profile because they have clustered together. And all these wild types have a similar expression profile because they have clustered together, except for wild type 2. But the wild types, in comparison to the knockouts, have a different expression profile, which is very evident because they are so much farther apart across PC1. So, two questions PCA promised to answer. One was whether we can actually find some interesting difference. It looks like we can. And whether we should exclude some of the samples from further analysis. It looks like we have to exclude WT2. Does everybody understand how you interpret a PCA plot in terms of your RNA-seq analysis, and how you can use it as an exploratory or a diagnostic plot in terms of even before you begin doing any statistics? Yes, people understand the inferences? Okay, I see one thumbs up. Anybody else? I see a yes. Okay, if no more questions, then
Yeah.
finally, yes, you have a question, Shubhajit?
Yes. Yeah.
Okay,
Can you repeat
let's do one thing, Shubhajit. Let's quickly finish first, and then
Okay.
we can
Sure.
stay back and answer the question, okay? Because yeah, let's not keep the others waiting, okay? Okay. So the next step now is, sorry, was somebody trying to say anything? Okay. So the next step is actually our differential gene expression analysis. Okay. Here, basically, what we do is we basically figure out which are the genes which are significantly different, right? So we basically try to figure out the group means. That, for example, if we have 3 samples of group A and 3 samples of group B, for each gene, we figure out a mean of group A and a mean for group B of that gene, and we try to figure out whether the difference in the group means is statistically significant or not, right? This might sound very similar to some statistical tests that you might have done, right? If you can remember, if you can go back to those times, you might remember something known as the student's t-test, that actually compares the means between two groups and figures out whether there is statistical significance or not. The idea is pretty much the same, but the tools or the tests we use are a bit different, and we will understand the math behind it. We'll understand how we figure out statistical significance, how we figure out log fold changes, which is basically the difference between the two. All of those we will understand. T-tests, by the way, are also used in differential gene expression analysis. Again, that is also something we will take up a little bit later. But the idea is to figure out a mean value for group A and a mean value for group B, right? And any deviations from the global mean would basically tell us that there is significant difference or not. Okay. The common tools or packages to perform all these kinds of statistical analysis are DSEQ2, 𝐏𝐥𝐸, and 𝐬𝐮𝐬𝐜𝐫𝐢𝐛𝐞. So 𝐏𝐥𝐸 is actually built on 𝐬𝐮𝐬𝐜. So we will be covering 𝐏𝐥𝐸 and DSEQ2 both, as they are the most state-of-the-art packages that are used these days. Apart from that, we can also visualize our results using plots such as volcano plots, heat maps, PCA plots, etc. We will also be seeing those in the upcoming lectures. And another very important thing that we'll be seeing in the upcoming lectures is something known as adjustment for multiple testing, or FDR correction, or false discovery rate correction. One of the techniques used is known as the Benjamin-Hochberg correction. Okay, so this is done to actually make sure that whatever observations or whatever significant differences we are observing, they are not just happening by chance, and there is actual statistical significance behind them, right? So this is actually something that helps us generate the p-value for the differences. Okay, and as you all know, in research and in statistics, you are only as good as your p-value, right? So that is what this is all about. And finally, when we have a list of differentially expressed genes, we can choose to do an n number of different things. What we will be focusing on in this internship is something known as functional analysis, where we will group the genes according to their biological functions and pathways and figure out what are the particular biological pathways and functions which are getting differentially disrupted in our condition as compared to the control. Okay, so these are some typical plots that we get out of them, right? As you can see, on the y-axis we have the names of the pathways, and on the x-axis we have something called a gene ratio. We'll understand what the gene ratio is. It is important to understand. The size of the plot, basically the size of the point basically tells us the number of genes in our list that correspond to this particular pathway, and this is nothing but a p-value associated with this pathway being differentially expressed or not. Okay, so the tools that help us do this are some things known as David. There is something known as GSEA, which stands for Gene Set Enrichment Analysis. There is something known as enrich-R. These are all R packages or UI tools which actually help us perform these analysis. Okay, there is something called 𝐏𝐥𝐸, which is nothing but a pathway database. There are certain databases that help us perform this analysis as well. So these are all things that we will go through during the end stage of our internship, when we actually I think we have a session on functional enrichment. This is when we will actually use all of these to actually figure out the biological differences in our data with respect to the pathways and biological functions. I will show you guys some easy tools and using R, how you can actually perform all of these analysis. Okay, so with that said, the key takeaways of today's lecture are that a good experimental design is very crucial. You need to decide on a proper number of biological replicates, controls, library prep, etc. Sequencing only generates raw reads, not insights, right? That is why it is very important to maintain high data quality, and your data quality will determine all downstream analysis, right? Third point is reference genome indexing enables fast and accurate alignment, helps in saving a lot of time and computational intensity. Okay, quantification, that is converting the reads to the expression values. That is what is the crux of differential gene expression analysis. Alignment is not the only step. You also have to quantify. You also have to figure out how many reads are assigned to each gene, right? And differential expression basically identifies biological signal, right? There are statistical tools that help us distinguish true biological changes from actual noise. Okay, and functional analysis basically tells us and translates the data into the actual biology. As I have been saying, your analysis is only as good as the biology. Okay, if your analysis does not make sense biologically, your analysis will not hold up anywhere. Okay, you are a biologist first and an analyst second, so our analysis must make sense on a biological logic. Okay, it should make sense. So with that said, we have come to the end of our second lecture on the fundamentals of RNA-seq. From the next lecture, we'll start with a little bit of Linux basics. I will share the manual to install WSL, or the Windows subsystem for Linux, for Windows users. For Mac users, it is absolutely fine. You can use your normal command prompt. It is almost the same as Linux's. And if you're already using a Linux system, then all good, no problem there. Okay. And if you have any doubts with respect to the installations, please feel free to reach out to me on Telegram. I will be solving doubts of related to any technical issues on Telegram. We will not entertain them during the class because it really takes up a lot of time. Okay, so I will share the instructions tonight only. Our next class is on Monday, so you have the weekend to figure out the installations. Please do them before the class, because otherwise, if you waste your time installing stuff during the class, it will be difficult for you to follow. Okay, so I would suggest over the weekend, perform the installations. If you have any doubts, reach out to me on Telegram. Okay, with that said, I think Bhavni, we can wrap up.
Yes,
Shubhojit,
Suryodaya.
Shubhojit, you had a question, I remember. I would request you please post it on Telegram. I will answer it to you before tonight, before the end of the
Yeah,
day. Okay.
yeah, sure.
Okay. Okay.
Thank you, Suryodaya. Dear participants, please fill the feedback form. It also acts as an attendance form. So I'm requesting everyone to fill it out. And as mentioned by Suryodaya, for the next couple of days, we don't have session. Next session is on Monday around 5:30 p.m. Indian time. I'm requesting everyone to join on time. Those are present here and also going to review the recordings. Please join us from next session. We won't entertain those who are not joining. With this, let's wind up for today, and you will be receiving this recording link in a minute or two. So please review it in your weekend. Thank you all. Thank you once again, Suryodaya.
If anybody else has any questions with respect to the lectures, please feel free to reach out through Telegram. Okay.
I'm not sure how to join the Telegram group actually.
Bhavni will be able to
You
help you
might
with that.
have received today in today's mail. You might receive the link to join the Telegram. Kindly join through the link. Is that clear? Ruby, is that clear? Yeah. Thank you. Fine.
Thank you,
Thank you, Suryodaya.
everyone. See you again on Monday.